What you see here is the UCA, the Catholic University, and a part of the River Plate facing the other shore of the Rio de la Plata. First of all, I want to thank Leonel for inviting me to this extremely interesting workshop. I am enjoying very much this series of lectures by real expert colleagues. Now, my talk today deals with a very specific topic, and I usually forget my younger colleagues. This is the last pre-pandemic photograph of my smallish group. The colors of the lettering are not arbitrarily chosen, as you can see here. There's a reason behind the choice of yellow and blue. The Rio Platenses will appreciate this. Now, my talk deals with the motion in the plasma membrane of one of the most abundant neurotransmitter receptors, the nicotinic receptor, which you see here in the neuromuscular junction. This is a peripheral synapse. This is a presynaptic bouton or nerve terminal, and receptors sit at the crests of these post-junctional folds. These are pentameric receptors, and they receive acetylcholine, the neurotransmitter, from the presynaptic terminal. If we were to look at this synapse at the postsynaptic side from the presynaptic side, wait a minute. All right, of the three possible motions that a receptor can undergo in a membrane, that is translational diffusion in the plane, wobbling, and rotation, I will only deal with the translational or diffusion in the plane of the membrane. As I was saying, if you look at the postsynaptic membrane in this direction, what you would see is something like this: a quasi two-dimensional crystal in 2 dimensions of receptor particles packed at densities of 20,000 particles per square micron. This is extremely high density, and of course, under such conditions, the receptors are essentially immobile in the adult neuromuscular junction. But this is not always the case. In the embryo, for instance, receptors are synthesized as monomers and inserted in the membrane as such. It is only after birth that receptors acquire this larger clustering or patching, as it's called, which is micron size, and this occurs even before the nerve reaches the muscle cell in postsynaptic day 30. What I call the socialization of the receptor occurs in a rather narrow time window, 1 day, 24 hours between embryonic day 13 and 14, when the receptor goes from the monomeric form to higher oligomeric aggregates. And I developed the idea that the aggregation phenomenon or clustering was initiated by receptor-receptor interactions. What we found is that cholesterol played a role in this process, and we identified, together with my younger colleague Javier Bayer, that each receptor subunit could bind up to 3 cholesterol molecules, predominantly in this anomalous region in the transmembrane domain of the receptor. So since the receptor is a pentamer, we have a total of about 15 cholesterol molecules per receptor macromolecule. And cholesterol interacts very tightly with some of the transmembrane regions. The total free energy of interaction of these 15 cholesterols amounts to nothing less than 40% of the lipid free solvation energy of the entire protein. So you can imagine how tight this is. Now, the physiological importance can be revealed by some experimental work we did. For instance, if you mutate and replace phenylalanine in one of these cholesterol recognition domains in the transmembrane region, you practically abolish the interaction with a cholesterol-containing monolayer. And NMR studies that we conducted with Tony Watts at in Oxford corroborated the affinity of the recognition sites of these transmembrane domains for cholesterol. Now, when were these tight contacts between cholesterol and ligand-gated ion channels established? And the answer is a very, very long time ago. In the case of prokaryotes, like primitive bacteria, it is hopanoids, some lipids which are extremely similar to sterols, which occur in eukaryotes, that appeared on the surface of planet Earth about 4 billion years ago, whereas in the eukaryote, it's these other forms of the cholesterol and related compounds that appeared in planet Earth. Now, which structural and functional aspects can we explore with microscopy? Both structural aspects in the fixed cells, for instance, we can study the population topography in static samples, imaging chemically fixed specimens, and we did this quite some time ago, 1 year after the introduction of the STET nanoscopy technique by Stefan Hell and colleagues. We published in 2007 the first study of a neurotransmitter receptor protein and the second membrane protein to be explored with nanoscopy. Here you see 1, 2, 3 CHO cells which express on its surface abundant number of receptors in confocal and in STET, which in those days we used to label it with the fluorescent probe ATO 532, and you can see it again in resolution that you obtain with the STET technique. Now, in this cartoon, you can see that the receptor is only 8 nanometers in width, and of course, this makes it brings us the conundrum that we cannot discern, or we couldn't in those days, between a single molecule to a few molecules within the radius of 60 nanometers with the resolution that we had 13 years ago. But nonetheless, this puts us in the realm of electron microscopy, of nanoscopy. Imaging receptors in monomeric or multimeric forms could only be accomplished by electron microscopy, as you see in this negatively stained specimen here. Upon returning from my sabbatical in Göttingen with Stefan in 2007, we were decided to build a nanoscope in Argentina, and we did so in 2008 with the help of Stefan and his 12 postdocs came to Bahia Blanca, where we were living in those days. And we built this type of microscope, not a STET because it was too expensive and I did not have the expertise to do this, but a STORM microscope, which is a single molecule localization microscopy type of scope. And here's an example of what you can see with a STORM microscopy nowadays. This is a single cell, but you can see the complete ventral surface patch with receptor molecules. We can count up to 90,000, 100,000 molecules per receptor per cell. This corresponds to the nucleus. And when you blow up a part of the cell, you can see that not only are there monomeric type in appearance single molecules, but also particles will look like supramolecular aggregates. And you can interrogate this with by these aggregates of molecules with Bayesian class metrics, from which you can, with this approach, you can look at the discern between single molecules and small clusters. You can establish, for instance, the number of particles per cluster, or the, as you see here, or the sizes and the percentage of material which is in cluster form, all the metrics which are relevant to the static type of imaging. On the dynamics, as you know, time is a key variable in biology, and this is also the case in understanding receptor function. We want to know how the receptor moves and why it doesn't follow a conventional Brownian type of motion. You could do it with STED microscopy, but the time resolution of STED for live microscopy is not always adequate for membrane proteins. And as I told you, building a STED microscope was beyond our budget, so we went for the single molecule approach. And to this end, we combined single molecules localization microscopies with single particle tracking, which is a good compromise in the time-space domain. As it was explained very nicely yesterday by Luca Lanzano and Fernando Stefani, in STORM, we look at the diffraction-limited wide field image of the single molecules, and you have to extract this information a posteriori in the computer, from which you reconstruct the centroid of the PSF, and from that you can reconstruct the entire object that you're trying to explore. And of course, as Luca Lanzano explained yesterday, the more photons you collect, the better resolution you obtain and the better your localization precision. And so with the STORM technique and with the PALM technique, we can address both the static imaging and the dynamics of receptors moving in the plane of the membrane. And the first paper published on this direction was by these two colleagues, two postgraduate students, in fact, of physics, University of Buenos Aires, Pablo Camino and Alejo Mosqueira, who are now in Spain and Denmark, respectively. So one question that you address is, for instance, population dynamics. We want to know the ensemble or population level. How do these receptors move? Do they move like all the particles in this cartoon? Well, the answer is no. They are not as disciplined as these particles, but rather move in different directions. And of course, the most conventional way to analyze these trajectories, individual trajectories at the ensemble level, is to follow them by constructing the mean square displacements of the whole population. And we next classify the trajectories, and what we see is a great heterogeneity of diffusional modes, from subdiffusive particles to superdiffusive particles, predominantly subdiffusive, as tested by looking at these two parameters, the beta or anomalous exponent and the K sub beta, which is the generalized diffusion coefficient. And I will talk later about the effect of cholesterol, but there is a subtle tuning of the diffusion of these particles by manipulating the cholesterol content of the membrane. At the individual level, at the individual behavior, we can also look at single particles, which don't always go against the current, as in this graphic metaphor. And when we focus on the individual trajectories, we found something rather unexpected. All trajectories, not both, all trajectories from subdiffusive to superdiffusive alternate between free walks, here shown in red color, with unconfinement sojourns. The particle stops for some moment. The longer the trajectory spends in the confined mode, the more subdiffusive, slower it will be its trajectory. So the proportion of the confined sojourns, that you see here, determines the diffusional regime of the population. So the next question we addressed was, which physical model accounts for this type of behavior? And probably the CTRW, or continuous time random walk, is the most typical transient immobilization that the protein can undergo from binding to other partners. It could be receptors, it could be cytoskeletal elements, it could be scaffolding proteins in the membrane, or it could be the extracellular space. Again, with Alejo and Pablo, what we did is to observe experimentally this parameter, which is called the escape time. And this escape time increasing radii of the around the given particle. And this is typically observed in the case that you have a non-ergodic type of diffusion, which is permitted us to exclude the continuous time random walk type of model, because it doesn't follow this type of premise. So we next explore two alternative models that could account for the predominant subdifusal model. One of them, so-called fractional Brownian motion, emphasizes the viscoelastic properties, that is the restitutive forces that, like an elastic band, the membrane tries to impose on the particle diffusing across it. The other model, the so-called obstructed diffusion model, emphasizes the spatial heterogeneities or obstacles that the protein can face in its lateral diffusion. And here you see two examples. First of all, the way to test it is, or to differentiate between these models, is to look at the parameter which is called turning angle analysis, looking at the changes in the direction of the particle as it walks along the membrane in its trajectory. Here you see a typical example borrowed from the Diego Krapf and his group on potassium channels. Fractional Brownian motion tends to show this type of behavior of the turning angle. As we go to higher angles, the curves tend to flatten and plateau, whereas in the percolation and obstructed diffusion model, as you see here, the curves tend to go upwards for large angles. And this is precisely what we found in our experimental data. So we favor this type of obstructed diffusion model. And what type of particle or what type of object could produce such an effect? One of them is these small actin corals. These corals could encircle and circumscribe the motion of the receptor protein in the submembrane actin network, producing important obstacles to receptor diffusion. And in fact, early studies by Virginia Borroni and Jorge Wentz in my lab in 2010 had already shown by STED technique and manipulating the cytoskeletal integrity using cytochalasin D or just vatinolide, which interfere with the cytoskeleton, that the sizes of these domains varied a lot and interfered with receptor distribution in nanoclusters. Now we went to one step further and we dissected the two portions that I mentioned before. We could separately analyze the confined portions and the free diffusion portions of the receptor trajectories, and sure enough, the obstructed diffusion model fitted nicely the confined portions, whereas the unconfined portions follow the free diffusion type of behavior of this type of analysis. So I'm just about finished. As you can see here, this indicated that it's the confined part of the trajectory that dictates the shape of the compound curves. Now, to our surprise, when we apply the analysis that Ibrahim Cissé and co-workers developed at the MIT when Cissé was there until this year, for transient self-aggregation phenomena in the nucleus, when we applied to the receptor protein, we observed that there were confinements much larger than what I have just discussed for the confinement sutures, 4 times larger in area and several seconds longer in duration. And this could be were extremely cholesterol dependent when we applied beta cyclodextrin to the membrane, and we extracted or we replenished cholesterol to the membrane. We observed that there were statistically very significant difference between this type of confinements, which of course affected the population. It was in the order of a few, almost a micron in size in some cases. Now, this morning you heard Federico Lecumberri speaking about deep learning, so I decided to add a few words about this. We have recently, this month, published a paper on deep learning approaches as applied to static microscopy images, and now we have another one on dynamic data. And with Hector Buenamaison, a graduate student in informatics, we challenge these models of behavior, of diffusion, developing a concatenated type of networks to select the best physical model, the same that we explored in the previous slides. And for instance, it took us 21 days to train the system using a relatively fast PC and an NVIDIA Titan V graphic GPU, as we heard this morning. And sure enough, with the deep learning approach, we were able to confirm the goodness of the two-state model from Grevenkov, and we could corroborate the predominance of subdiffusive diffusion over Brownian and superdiffusive. And we could also look at the effect of cholesterol, which was very marked on both bungarotoxin label and monoclonal antibody type of labels on the receptor protein. So in conclusion, an important percentage, I didn't go into this for reasons of time, almost 50% of the receptors are immobile during the entire time window of the SP single particle tracking recording. The mobile receptors exhibit a highly heterogeneous behavior, subdiffusive, Brownian, and seldom superdiffusive. The best model to fit this mobile population is a two-state ergodic model with intermittent exchange between free Brownian diffusion and confining sojourns. 30 nanometers radius is the average, and half a second duration is the lifetime of this period. And in addition, the CC analysis revealed larger areas, average 150 nanometers radius, and longer lifetimes in the order of seconds, which might constitute the physical self-obstacles. That is to say, the self-aggregation of the receptor that impedes the trafficking across the membrane. So a conclusion of our current choice of model is that in a minimalistic cell system devoid of anchoring proteins, as we use in the CHO system in the lab, suffices to, appears to support the notion that the receptor self-aggregation suffices to produce nanoclusters, and this event that takes place in the embryo, the elementary state of the basis of synapse formation, the transition from monomers to aggregates, small aggregates or nanoclusters of receptors, the beginning of the social life of the receptor. So I finish with this. Please do write to me if you have interest in the type of work that we are doing, or if you have some doubts. I will provide a reading list later on.